Hi, this is Dr. Cameron Sheeler with UAB's biotechnology program. Today we have a saga for you. It is called Transformation, the saga of a plasmid and E. coli. Will they meet? Here's today's plasmid. It's from Adgene. It's the PEGFPC1ER alpha, circular piece of DNA, our plasmid. It's 6,523 base pairs long. But the important aspect of what we're going to use today is on this circular plasmid is a resistance. It will make resistance to neomycin and also kanamycin. We can use that as selection markers. That is, how are we going to determine which bacteria have incorporated our plasmid? That's what we're doing, transforming E. coli with this plasmid. Let's go to the lab, transform, and select, and have a grand time. Woo. The E. coli that we're using is from Invitrogen. It's called One Shot Top 10. These are competent bacterial cells that are primed and ready to take up the plasmid. So I already have some thawing out. Igor has already done this. I take the vial out and put it on ice, let it thaw out for 30 minutes. So our first player, E. coli, is waiting for us. And we have to get our second player, the plasmid. I have my frozen plasmid right here. And we'll put this on ice. The saga is ready to begin. Will the plasmid meet the bacteria? Will the bacteria embrace the plasmid? Will the plasmid go into the bacteria? This is today's saga. Let's find out how it ends. Our bacteria is thawed out and our plasmid is thawed out. We are going to quickly centrifuge to bring it down. Now, I deviate from the manufacturer. I teach a 2-hour class, so I've modified a little bit. Here's the deviation. This has approximately 50 microliters in here. What I do, I am going to transfer 25 microliters from this tube into a new Eppendorf tube. I'm going to use this as a control, or if you wanted to use this also, we can actually add plasmid to this. Here's your homework. What will happen to that bacteria that does not have the plasmid? And I plate it on a plate that has a selection, kanamycin. What do you think is going to happen? We'll find out as our saga continues. Our plasmid, I'm going to add 1 microliter. You can actually add 1 to 2 microliters in there. When we do this, I'm going to flick it real fast, centrifuge it, and then we're going to incubate it on ice. There, put it in, mix it like that, quick centrifuge. Now we incubate on ice for 30 minutes. During this time, the plasmid will come into contact with the competent E. coli. However, we're setting it up for the plasmid to go in. We'll come back in 30 minutes to see what to do next. We have let the plasmid and bacteria incubate on ice for 30 minutes. That allows the plasmid to come in contact with the competent bacteria. Meanwhile, we have to set our water bath to 42 degrees Celsius, and you'll definitely have to have an ice bucket. Our control, no plasmid. Plasmid. Let's go ahead and heat shock for 90 seconds at 42 degrees. Next step is we actually have to add our SOC. Dr. Sheeler, what is the SOC? I knew you were going to ask that. SOC, super optimal broth with catabolite repression. SOC. Now we're going to add 250 microliters of SOC. To the plasmid with E. coli. Turn this off. We're not done. Now we're going to put it in a shaking incubator set to 37 degrees Celsius. And I have RPM set at 225 for 30 minutes. The original protocol was for 1 hour, but again, I have a 2-hour lab that all this has to get done in 2 hours. We will put a 50 ml conical tube in here, drop in our bacteria and plasmids, shut it for 30 minutes. This is the recovery stage. The bacteria has been through a traumatic experience with that 42 degrees Celsius. Now, we're going to let it recover. It's this rich media. It's going to help start making its antibiotic resistance. It'll help start making the proteins, anything it has to do to recover and start growing. We'll be back in 30 minutes. Time's up. Okay. I'm going to do a 1 to 10 dilution of this transformation mix, and I'm going to put it into LB media. Mix it up just a little bit. I'm just going to mix it up, mix it up, mix it up, mix it up. All right, now we're ready to go. We set this to 50 microliters, and you'll see what I'm going to do right now. I've got plates set up here. Control. These plates are LB auger plates with kanamycin at 50 micrograms per milliliter. That's the concentration. And you're going to say, why do you have kanamycin? Again, we're trying to select for any bacteria that have incorporated the plasmid. Fresh spreaders. Grab one, and I'm just going to add 50 microliters. You can add 100 of the undiluted. Just kind of put it in the center. Now I'll take our sterile spreader. Here we go. Just kind of move it around. Spread it out. Turn it back upside down. When we plate, we put everything on the bottom of the plate that has the auger. When we incubate it overnight, it helps prevent contamination, and what it will do is just grow. Now we're going to put 50 microliters of our 1 to 10 dilution in here. The point is we want 1 colony. We only need 1 colony. So I'm actually diluting it out just a little bit so that I don't overwhelm it. Making sure we spread it out. Now, I'm going to add a little bit less, 25 microliters. There's multiple different ways to do this. I'm just adding less volume. Add to the center. You have to use a spreader, brand new one every time. Now, the control. Remember, this was the remaining bacteria that we did not add a plasmid to. This is still a LB auger with kanamycin in there. That's good enough. Upside down, and we're going to put it into our incubator at 37 degrees Celsius. We will incubate this overnight, and tomorrow we'll see how our saga ends. Will we get bacteria growing as individual colonies? Will the control have growth or no growth? We'll find out how this saga ends. I fell asleep at my desk again. Ooh, Igor brought my coffee. Let's go find out our results. All right, it's been overnight, and now we're going to find out if we have growth or not, and we'll find out if our negative control has growth or not. Ooh, the saga continues. First, I like to check my control. Nothing growing on the plate, which is good. If we had something growing on the plate, that means our kanamycin was not working. This is a good sign. Ooh, 50 microliters. Look at all those beautiful colonies. Sometimes if you have so much bacteria, you can't pick 1 colony. So that's great. If I had to, I could pick 1 colony from here. Now these are 1 to 10 dilution samples. This is 50 microliters of 1 to 10, lots of colonies. Less than the undiluted 50 microliters. and 25 microliters of our 1 to 10. The last colonies, it's easier for us to pick a single colony. Igor set up 10 mils of LB media, and it has 50 micrograms per mil of kanamycin. Remember, we're selecting for bacteria that have been transformed, have uptaken the plasmid. And we're using kanamycin as our selection. We're going to pick 6 colonies. As you can see, the colonies are quite separate. All we have to do is find one. All you're doing is touching it. Boop. Open this thing up. Drop. That's it. Let's pick another one. Don't put the cap on too tight. Have it wiggle, so basically wiggle, wiggle, doesn't come off. And then we're gonna tape it. Don't tape it all the way down. We want air to get in. I'm kind of with my thumb pushing it up. Just pushing it up so it's not being pulled down, and just put the tape on to hold it in place. See, still moves. Put it in our shaking incubator. Now we just let it shake overnight. Tomorrow, we can use each of these tubes for plasmid isolation. This concludes today's saga. The bacteria took up the plasmid. We're able to isolate individual colonies. The negative controls work beautiful. I just love happy endings. If you have any suggestions for future sagas, please email me at the below address, and maybe we'll pick your saga. This is Dr. Sheeler signing off. You have a great day.